In this section, we're going to explore ways that we can use physical methods to control microorganisms. In our next video, we'll get into chemical methods. When we look at physical methods, heat, of course, is going to be the #1 physical method that we have for controlling microbial growth. We'll also look at radiation, which we touched on a little bit when we were looking at DNA and mutations. We'll also talk about filtration and desiccation. So heat is really the most common. It's the oldest method that we have in terms of microbial control, and every time we go into the lab, we use heat. We use our burners to sterilize our loops before we begin our experiments. So heat, of course, one of the easiest forms also of microbial control. Heat kills microbes by altering their membranes and also denaturing proteins. Now, when we were talking about DNA and the genes and proteins, and I mentioned in one of those videos that the structure of a protein determines its function. That, you know, if we had an amino acid that was in the wrong place, or we were missing an amino acid, then that protein is not going to function as it should. Denaturation or denaturing of proteins is when we alter that structure. We alter that shape of the protein. If we alter its structure in any way, we change its shape or we interfere with some of those amino acids, then that protein can't work anymore. And heat is one of the ways that we can denature proteins. For instance, if you cook an egg, so you break that egg out of the shell, you put it in your frying pan, and you cook that egg, you can't ever take it back into that liquid form that it was in when it first came out of the shell, because you've denatured those proteins in the egg. And so heat will affect the structure of proteins. We have a couple of definitions here: thermal death point and thermal death time. The thermal death point is the lowest temperature at which all microbes are killed in a 10-minute exposure. Okay, so the thermal death point is the lowest temperature at which, if you apply that heat for 10 minutes, it's going to kill all microbes. The thermal death time is the length of time needed to kill all microbes at a given temperature. So if your temperature was, 50 degrees Celsius, your thermal death time would be how long the microbes have to be exposed to that 50 degrees Celsius temperature for them to all die. When we look at heat, we can use dry heat or moist heat. Dry heat would be like we do with our loops. Moist heat is when we use steam. So this would be like what happens in an autoclave, for example. Now, different methods of heat will achieve different goals. Some heat can be used as sterilization. Some heat can be used just as disinfection. It really determines what protocols you're using and the types of materials that you're trying to sterilize or disinfect. Autoclaves use moist heat, so they're using steam. Basically what happens in an autoclave is they use pressure to raise the temperature above the boiling point of water. When we alter atmospheric pressure, barometric pressure, we can alter the boiling point of water. Normal boiling point of water at sea level with 1 atmosphere of pressure is 100 degrees Celsius. But in an autoclave, we can manipulate the internal part, the internal chamber of an autoclave, to increase the pressure. And as we increase the pressure, it raises the boiling temp of water. Standard operating temperature for most autoclaves is 121 degrees Celsius. So we can achieve higher temperatures by adding pressure, which is usually about 15 pounds per square inch. When we look at sterilization using an autoclave, it depends on what material we're using and how much, but it's usually about 10, 15 to 20 minutes. Smaller autoclaves don't take as long because they can get up to a higher pressure and a higher temperature more quickly. So we typically don't have to leave things in the smaller autoclaves as long. This autoclave in this image here is a giant autoclave. So there would more than likely be a lot of probably surgical tools in this autoclave. And so it's going to take longer to get to the correct temperature and pressure. So the time that it takes to sterilize is going to be a little bit longer. Those tools are going to have to be left in the autoclave a little bit longer. The goal with the autoclave, though, is that we can achieve complete sterilization because the temperatures get high enough that we can also kill the endospores of bacteria. Another form of heat, or another type of using heat, would be pasteurization. Pasteurization is the goal of killing pathogens and also reducing the spoilage microbes, but it doesn't alter the taste of the food product. So we're maintaining food quality, but also being able to kill the majority of the microorganisms. Pasteurization has been around, of course, for a long time. Louis Pasteur first developed this technique for wine, but now we apply it for a lot of different beverages, including milk and juices. We have different forms of pasteurization. One is high temperature, short time. So this HTST, and you can see there it's 72 degrees Celsius for 15 seconds. It's a much higher temperature than traditional pasteurization, but it's at such a low time, only 15 seconds, a small amount of time, that it doesn't alter the quality of the milk. So it can kill bacteria, but still preserve the flavor of milk. Another type of pasteurization is ultra high temperature, or UHT pasteurization. This is 138 degrees Celsius for 2 or more seconds, usually less than 5. The goal with the ultra high temperature is that it's pretty much sterilizing the milk or other beverages. It may cause slight changes in the quality. It may affect the taste a little bit. But with ultra high temperature sterilization, you can have, dairy products, milk especially, that doesn't have to be, refrigerated. It doesn't have to stay refrigerated. So in areas without electricity, or when you have, natural disasters or things like that, the ultra high temperature sterilized milk can come in handy. If you ever go to a restaurant and, they have the little bowls of the half and half or the cream for your coffee just sitting out on the table, and you think, that should be cold. Well, if you'll take a little closer look at the labels, you'll often see this acronym here, UHT, on it. And that allows that cream or that half and half to sit out on the table without refrigeration, and it's not going to harm you at all because it has been sterilized. Now, when we switch gears and go to cold, so we go to the absence of heat, to refrigeration and freezing. Exposing microbes to low temperatures can be used as methods of microbial control. But of course, remember our psychrophiles and our psychotrophs, they can survive in cold temperatures. Refrigerators that are used in home kitchens are usually somewhere between 0 degrees Celsius and 7 degrees Celsius, usually somewhere between 35 and 40 degrees Fahrenheit. Refrigerators should always be kept lower than 40 degrees Fahrenheit. I would recommend that everyone get a thermometer to keep in their refrigerator just to make sure that you're staying within the correct temperature range. Now, at those temperatures, microbial metabolism is inhibited. Okay, so it's slowing the growth, but it's typically not killing the microbes. That's why you put your leftovers in the refrigerator, but eventually, leaving those leftovers in there, they will spoil. You know, you're slowing down the growth, but you're not killing the microbes. They are still growing. So, you know, leftovers shouldn't be left in your refrigerator more than 5 to 7 days. Freezing can stop microbial growth and may even kill susceptible organisms. And this would be below 2 degrees Celsius. So we're actually at a point below the freezing point. The bacterial growth that is slowed down or stopped during freezing can pick back up in thawed foods. And so, you know, you think about thawing, you should only thaw things in the refrigerator or in cold water or in the microwave. You know, just sticking something that's frozen in the refrigerator and then letting it completely thaw out, that may not have killed everything that's in there, and that bacteria can start growing again. We can actually store bacterial cultures. They're frozen at temperatures of negative 70 degrees Celsius or below. And they can be in, you know, dry ice or frozen using liquid nitrogen. But this is actually usually a form of preservation. In fact, some of the samples that I get have been frozen to preserve them. And as soon as we put them in the refrigerator and basically reconstitute them and put them in the incubator, they'll start growing again. As we saw with the autoclave, we know that we can alter pressure. And, you know, with the autoclave, we're using pressure and heat, but we can use just pressure to help control microbial growth. High pressure processing or pasteurization can be used in the food industry. It preserves taste, but it will kill bacteria and other forms of microorganisms. It kills all vegetative cells, but those endospores may survive. So this is not sterilization. And mostly used for food or tissue. We can use hyperbaric chambers where we put a patient into this chamber and they breathe in pure oxygen, which is at a higher pressure than atmospheric oxygen. And this basically increases oxygen saturation in their tissues. And for our anaerobic microorganisms, they can't withstand that. Remember that oxygen is toxic to them. So by putting the patient in this type of environment, the high exposure to oxygen can actually kill these microorganisms. But that's really our only uses that we have for pressure. We can't really do it for fomites because it, you know, when we put them in the autoclave, it's the high temperature, not the pressure. So pressure more common in the food industry and also on skin or other tissue. We can use desiccation or dehydration when we draw all of the water out of a particular, typically food substance. All cells require water, and if we take away all of the water, then we can control microbial growth that way. You know, we've used drying or desiccation for years. You know, before we had refrigeration, you know, farmers would kill their cattle and they would use salt to draw out the water, and then they'd hang it up basically to cure. And what was happening there is that the water was being drawn out. The water content in food which is called the water activity, can also be lowered without drying by adding salt or sugar. And that's how the concept of like honey or jams or jellies work, that we put enough sugar in there, then bacteria don't have enough available water to grow.